Hi, ladies and gentlemen. If you need a little help on the Evidence for Evolution lab, I just want to walk you through how to complete some of the lab activity. Hey, first of all, we've got some fossils here. You are going to be looking at the fossils, first of all, looking at the skulls from modern horse all the way back to its an early ancestor. So you'll be analyzing the structure of that skull, and then you're looking at the bones that make up the legs of the horse, and kind of investigating that from, again, modern horse back to its early ancestor. From here, those of you guys that are remote students, you will move on to our next activity. You're choosing one of the four here. So you're going to this website and choosing one of the four to explain what the pieces of evidence tell us. So choose either past lives, geologic time, paleological, or biodiversity. Choose one of those four. I don't know why that's repeated there. Then you get to the next station, whether you're a remote student or in person, you're going to read through this information about biogeographical evidence. It goes through and explains what it is. Then it talks about camels as an example, island biogeography, and then a specific example of evolution that was observed in the 1930s. On the next slide, you're going to use the information from this one to help you answer the questions here. So you're answering these questions. I am going to throw out, because they didn't really provide you guys with the info to answer question number 2. So you guys can remove that question. But the other ones you should be able to answer. Then we move on to looking at anatomical evidence, and you're comparing butterfly wing and bird wing. How are they different? And then we've got a cavefish and a minnow. How are they different? And then assuming the two came from the same original ancestor, right? So if they had the same ancestral species, why did one evolve like the cavefish and the other one evolve like the minnow? When we get to the anatomical evidence, if you're a remote student, you are going to use arrows, or you could put boxes around each bone. You'll see here that the human arm is labeled. So we've got the humerus. You're going to find the humerus on all of the other organisms as well. So it's the top bone, and the bones go in order. First bone is humerus. Next two bones are the radius and the ulna. Radius is going to be on the thumb side. Then you've got the carpals, the wrist bones. They tend to be a small cluster of bones. And then the first bone after the carpals, so in the bat, it would be this long, skinny one here. That would be a metacarpal. And then the last bones, in this case, there are two of them in the bat, are the phalanges. You'll see in the crocodile, here are the metacarpals. They're the first bone after the carpals, and then there are two bones, and then there are 1, 2, 3 bones. So you might see a variety of phalanges, and some of them will be longer, and some of them will be shorter. Then you explain what type of movement these different bones allow for in the organism. So like a bat, these bones allow the bat to fly. So that would be the type of movement here. And then you would compare to the human form. So you would look at the bones in the bat and say, okay, the phalanges are much skinnier and longer in a bat than they are in the human. And the humerus is shorter. Same thing with the radius and the ulna. And the carpals are a little tinier. And then you're explaining the function. So again, in terms of humans, right, we're using our arm to move things or to pick things up. A bat is going to use these bones to allow it to fly, to flap its wings. So you just describe the difference in those two types of motion here in the last column. Then we get to some questions about homologous, analogous, and vestigial structures. Homologous structures are structures that have the same basic bone structure, okay, but they function differently. Okay, so they had the same evolutionary background where they came from, so their structure is similar, okay, but they have different functions. Analogous structures are structures where the function is the same, but they actually have different evolutionary backgrounds. So they're not, the structures themselves are not actually related to each other. So an example of that would be like a bat wing versus the wings on like a butterfly, right? So those two structures would not be the same in terms of their evolutionary development. Okay, so they're analogous because they end up having the same function. And then a vestigial structure is a structure that is no longer being used. Okay, so it's a structure that an organism has, but it doesn't actually do anything for that organism. So an example of a vestigial structure in us would be our appendix. Okay, the appendix is part of our digestive tract, but it doesn't actually do anything. It doesn't help us digest any food. So we believe it's a remnant from an ancestral species that needed to be able to digest different types of food than the foods we eat today. Then you move on to embryological evidence, right? These are developing organisms. Okay, there are 6 different organisms here. You're just making an educated guess on which one you think matches with each species. So you'll just put the letter here. There's no wrong answer here. You're just trying to look for, are there any maybe differences that might lead you to think one is related to one organism or another? You'll continue that same process. These are older embryos, okay? And then you'll have some questions here kind of in a more advanced stage shortly before birth. All right, ladies and gentlemen, those of you guys that are at home, okay, you guys will then do the molecular evidence. Okay, so it's in a slightly different order for those of you guys that are in person. Okay, when you're doing the molecular evidence, you're going to highlight or put a box, okay, around any amino acid. We're comparing the amino acid sequence for a specific protein called cytochrome C. We're comparing the human sequence to the sequence in the other organisms. So I look at each column. First column, all Qs, so there's no differences. Second column, all A amino acids, so there's no difference. The third column, okay, we have P as the amino acid, okay, and there are differences in this column. So I've highlighted those differences, okay, and you would continue that through every column. And then once you've done that, and by the way, this last one, see there's a dash there, that counts as a difference. That means that it's actually missing an amino acid there. Then when you get to the next slide, you're then going to tell me how many differences were there in horses compared to people. So you would look across the row for horses, count up how many total differences there were, and you record that number here. More differences, more distantly related. Okay, and then the second question here, do any organisms have the same number of differences from humans to in cytochrome C? And remember, cytochrome C is one common protein, so one protein that's in common between all these species. There are lots of common proteins, so maybe if we have the same number of differences, we might investigate a different protein and use that to help us to distinguish which one's more closely related. All right, ladies and gentlemen, good luck on that lab. Let me know if you have any questions. Feel free to come see me in Zoom or send me a chat message.